Hello, everybody. This is welcome to the today's seminar or webinar. On behalf of the SingHealth Duke-NUS Global Health Institute, I'm delighted to welcome all of you to this first of a three-part webinar series, which is titled COVID-19, Looking Back and Looking Forward. So at the moment, the world's attention is understandably focused on how to manage the current pandemic. And in this webinar series, we want to shift the focus a little bit to look at the broader question of why such a virus originated in the first place. So in these seminar series, we'll describe current frameworks and strategies that help identify risks for pandemic emergence, and also the current tools that we have that can be used to mitigate these risks. And we're posing the question of how can we use our understanding and available tools to prevent future occurrences. So today's session is the first seminar, and we're looking at the role of evolutionary and ecological factors in pandemics like COVID-19. And in this seminar, we'll walk you through the web of interlacing factors that have led us to the situation that we're in today. And also please look out for information on subsequent sessions. We've already sent out the invitation for next week's seminar. So today there are two speakers. There's myself and James Rudge. To briefly introduce myself, I'm a professor of emerging infectious diseases and global health at Duke-NUS and the research lead at the SingHealth Duke-NUS Global Health Institute. The first speaker today is Dr. James Rudge, who's an Assistant Professor of Infectious Disease Epidemiology in the Communicable Diseases Policy Research Group at London School Hygiene and Tropical Medicine. And he also has an adjunct professor position at Mahidol University in Bangkok, where James is based. James's research focuses on the ecology of emerging endemic diseases at the human-animal interface and the capacity of health systems to respond to emergencies. And he is currently leading a project on zoonotic and pandemic risk in relation to swine production systems in Cambodia, that it is my pleasure to work on that project with him, and James and I have been working together for a very long period of time. Now, before joining the London School of Hygiene and Tropical Medicine in 2009, he completed his PhD and Masters in Epidemiology and BSc in Microbiology at Imperial College London. We were having some connection problems earlier on. I'm not sure, James, if you are now able to join us.
Hello, can everybody hear me?
We can, James. Excellent.
Great. Thank you very much, Gavin. Sorry, I've been having connection problems, so I've joined on my phone, and yeah.
Okay, so if everybody could just bear with us while we arrange the screen sharing for James's slide deck, please. Okay, thanks everybody for your patience. So James, please take it away.
Great, thank you very much. Yeah, so I'm going to give a largely kind of epidemiological and ecological perspective on the drivers of pandemic emergence. So I'll go through some examples of ways in which environmental factors and so on can lead to the emergence of new diseases and pandemics. And then I'll move on to some types of studies and variables that we kind of need to get better information and data on to help better understand the dynamics of diseases at the human-animal interface. So next slide, please. Okay, so before going into this, I thought it's perhaps makes sense to briefly go back to the origins of communicable disease in general, which is of course closely tied to the emergence of agricultural societies during the Neolithic era about 11,000 years ago. So prior to this, of course, hunter-gatherer type human populations existed in groups which were too small to sustain and the transmission of contagious diseases. And so the emergence of agriculture led to more densely populated and sedentary societies in which disease transmission could be maintained. And also, of course, domestication of animals provided ample opportunities for exposure to zoonotic diseases and disease vectors. So in recent decades, we've seen significant ecological and demographic changes which are driving the emergence and spread of new diseases. And in some respects, these factors kind of echo the huge changes that occurred with the origin of agricultural societies. Next slide, please. So also, just to go back to basics for a minute and mention the epidemiological triad, which is among the simplest models for infectious disease causation, but also one of the most broadly applicable models. And it consists of the three main components of the host, the environment, and the agent. And I think this is probably a concept that most people are familiar with, but I think it just sort of captures at the most fundamental level the key importance of interactions between these things and the role that environmental factors, including ecological changes that we'll explore later, can play in disease emergence and spread. So the environment, of course, affects host distributions, densities, and interactions, and so on. But they can also directly affect the agent itself if we think about how pathogens may be able to survive or not, even outside of the host, which can be affected by things like climate, humidity, and so on. Next slide, please. So pandemics, as we all know, typically originate from agents that are circulating in animal reservoirs. So these might spill over into humans. Sometimes this occurs directly from the main reservoir, but it often also occurs via an intermediate host, which acts as a bridge between the natural reservoir and humans. So, of course, SARS coronaviruses, including the one responsible for COVID-19, are thought to have originated from viruses circulating in bats, but which infected and probably underwent further adaptation in other mammalian hosts before spilling over and transmitting among humans. Often these intermediate hosts may be taxonomically more similar to humans, providing opportunities for the agent to undergo further adaptation. I think we also shouldn't ignore the potential importance of transmission from humans back to animal hosts, especially if we think about things like influenza. So pigs tend to be susceptible to both human and avian flu viruses, and they also have their own flu viruses. So they can act as a potential mixing vessel for genetic reassortment of different virus strains and subtypes. So for many spillover events, transmission might end here, with humans being a dead-end host. But if you click the next slide, please. For some infections, we might then see limited human-to-human transmission, as has been observed for avian influenza viruses such as H5N1. So this occurs if the basic reproduction number is less than 1, meaning that on average, each case transmits the infection to less than one other person. You might still get some stuttering chains of transmission, but these eventually die out. But with each new infection in a human host, this provides a new opportunity for the pathogen to adapt further towards humans. So this stage can be crucial for pandemic emergence. And if the pathogen is sufficiently adapted towards sustained transmission in humans, this can lead to localized outbreaks and potentially the emergence of a new pandemic. Next slide, please. So emerging infectious disease events are thought to be increasing over recent decades. And this map by Public Health England just shows the wide range of newly identified infections, or ones that have spread to new geographic areas since only 1998, and most of these being of zoonotic origin. And while, of course, improvements in surveillance systems and capacity to detect new events is likely to play a role in this trend, it's unlikely that this is the only or possibly even the main reason. Next slide, please. So just before I go into some of these in a bit more detail, just an overview of some of the key drivers for the emergence or re-emergence and spread of infectious diseases. So environmental change, of course, associated with things like land use change, deforestation, and agriculture, forces wildlife populations to leave their natural habitats and have contacts with other animals and humans. Demographic and behavioral changes, including increased population densities, urbanization, and increased mobility, provide opportunities for infections to spread more easily once a new infection does jump into humans. Globalization and trade have not only massively increased the frequency and distance of travel of human populations, but also wildlife and livestock through trade, and potentially the vectors for disease transmission. Microbial adaptation and change is more the subject of Gavin's talk, I think, but this, of course, also plays a key role. And I won't really discuss this further, but breakdown in public health measures is nonetheless an important factor. For example, if we think about the re-emergence of vaccine-preventable diseases and antimicrobial resistance. Next slide, please. So, of course, it's not possible to predict exactly when or where a new pandemic will emerge with any degree of certainty, but we can get insights on global hotspots from looking at where events have occurred and the environmental and demographic variables associated with these. And this study did just this, fitting a regression model to covariates on demographic and environmental factors, and it just illustrates how this risk tends to be much more elevated in tropical regions with not just high biodiversity, but which are also undergoing rapid land use changes, particularly in parts of Southeast and East Asia, where many of these factors converge. Next slide, please. So, of course, in talking about drivers, I thought I'd start with wildlife trade and live animal markets, given the huge role these have obviously played in the emergence of the COVID-19 pandemic and also the SARS outbreak before that. So these are really mixing pots or melting pots of biodiversity, bringing together a range of taxonomic animal groups from different geographic regions, which may not really be in close contact with each other, if at all, in their natural habitats. And so really fertile environment for diseases to jump between species. And not only that, but the stress induced by the conditions in which these animals are held can increase their susceptibility to infection. Also, it's not just the animals on sale at these markets which pose a risk, but also scavenging animals attracted to the food sources generated, for example, by slaughtering and waste disposal practices at these sites, which can harbour additional pathogens or otherwise enhance opportunities for disease transmission within, to and from these sites. Next slide, please. And I think the emergence of Nipah virus in Malaysia in 1998 and 99, which then went on to cause further outbreaks in, for example, Bangladesh, is really a textbook example of how ecological changes, a combination of these, can lead to new emerging infectious disease events. So this was really driven by a combination of anthropogenic changes. So there was deforestation, which forced bats to forage for food in other locations. And there was plantation of fruit orchids around these intensive pig production farms. And all of these things led to increasing the interface between wildlife, livestock and humans. Next slide, please. And then, of course, livestock intensification. So on the left is just some plots showing how this is rapidly increasing over recent years in countries in Southeast Asia. And these are on a log scale, so almost exponential growth in some countries. So this, of course, has huge ramifications, particularly for things like influenza, for which poultry and aquatic birds are the main reservoir hosts, and also pigs play a role in the reassortment and mixing of different influenza viruses. So this is leading to a situation where we not only have these kind of small backyard farms with low biosecurity and high mixing of different species, but also these more intensified farms with high poultry and pig densities, which could potentially amplify transmission of a new strain of influenza, for example. Next slide, please. And then thinking about another disease of zoonotic origin, so Zika virus, which caused explosive outbreaks in the Americas in recent years. So this study just looked at a range of factors which may have led to which may have driven these outbreaks. And for example, they found that the prevalence of Zika virus disease was negatively correlated with forest cover. So just kind of supporting the evidence or the hypothesis that deforestation and urbanization potentially increasing the risk of Zika virus transmission. Next slide. Another vector-borne disease, dengue virus, and vector-borne diseases in general could be quite influenced by climate change. So this map just shows the estimated population at risk at the top in 1990 and the predicted population at risk of dengue in 2085 based on climate change factors increasing the distribution of the Aedes mosquito vectors. Changes in temperature may not only affect the distribution of the vectors, but also their reproductive and biting rates and the maturation of parasites and viruses within vector hosts. So again, just an example of how climate change can also directly influence the spread of infectious diseases. Next slide, please. So, of course, particularly in Southeast Asia, the population is rapidly increasing and has increased by over 30% since 1990. Around 50% of people in the region live in urban areas, and this is expected to grow to more than 70% by 2050. So urbanization is, of course, associated with changes in social structures and particularly increased personal mobility, which can not only directly affect the potential for a new disease to spread within human populations, but is also, of course, a driver for many of the factors we discussed earlier. Next slide, please. And I think this slide just kind of highlights how these drivers are not isolated phenomena. They, of course, interact in dynamic ways, and they're often mutually reinforcing. So, of course, population growth and urbanization are driving these land use changes and increasing demand for meat consumption, which impacts on our agricultural and food systems. And all of these can go on to affect the population dynamics of wildlife and livestock and their interface with humans. Next slide, please. So I think much of what I presented so far relates to quite circumstantial evidence, looking at trends and case studies or from statistical associations between ecological factors and outbreak events or disease prevalence. But to gain a deeper kind of mechanistic understanding of how these drivers can impact on disease emergence and spread, we really need to be able to better characterize the dynamics of infectious diseases within and between host species and also across the key stages of pandemic emergence that I mentioned at the beginning. So a key tool we have to do this is through mathematical models, which can then also help us identify strategies to prevent or reduce pandemic risk. But these models need to be informed by empirical data wherever possible. And although certainly not a comprehensive list, I just wanted to highlight a few of the key variables which we need to better understand and some of the approaches and tools that can help with this. So of course, disease dynamics in these host populations are affected by the population dynamics, but also the interactions between reservoir populations. And this can be informed by things like geospatial analyses of densities and distributions. If we're thinking about livestock populations, characterization of the livestock value chains and trading networks, and phylogenetic analyses of their pathogens can give insights into how diseases spread between these different populations. In terms of ascertaining the prevalence of disease in these reservoir hosts, increased pathogen surveillance is of course important, but I think a lot of surveillance and sampling so far has been geared towards, just obtaining samples for genetic and laboratory analyses, and maybe we need to give more thought to how we can better design these sampling strategies to really inform how the dynamics in these populations change over space and time. So that would involve spatially and temporally stratified sampling. And of course, next generation sequencing approaches are really crucial for identifying potentially pathogens that aren't well defined or not yet identified. Next slide, please. In terms of spillover dynamics, so these are of course affected by the prevalence in the reservoir, but also human reservoir contact rates, including their frequency, duration, intensity. So we can get better information on these from things like behavioral and contact pattern surveys, and this is something we've been doing in Cambodia in relation to pig production practices, and also again through spatial overlap studies through things like remote sensing data. It's also important to be able to identify groups which may be at high risk of zoonotic transmission. So this can also be informed by things like serological surveys, looking at which groups may have had high exposure to different zoonotic pathogens. The spillover dynamics are also affected by probability of transmission. So things like pathogen load and shedding studies in different animal hosts can be important for getting a better picture of how this may vary across different host species. And we need to get better insights into the key transmission modes and pathways which lead to these spillover dynamics. And of course, many diseases have several different modes of transmission. So things like environmental persistence studies can help us identify what the potential importance of kind of fomites or transmission through environmental reservoirs, and epidemiological risk factor analyses in relation to people's behaviors. Next slide, please. In terms of insights into human-to-human transmission, again, social mixing patterns data is really useful here. So actually, if you just go to the next slide, I'll quickly give an example of that. So we've been looking at collecting data on how rates and patterns of contact between different age groups in Cambodia vary between different settings, for example, within the home, at work, at school, and so on. And this kind of data is really useful when we, for example, for looking at the impact of non-pharmaceutical interventions like social distancing strategies in different settings for mitigating disease spread. If you could just go back to the last slide again, please. Things like individual variation in transmission. There's emerging evidence on the importance of this in the COVID-19 pandemic and the role of super spreader events. So this is something that we need to better characterize for different pathogens. Also, of course, the epidemiological and clinical features of diseases, including their incubation and infectious periods and the role of asymptomatic transmission. Again, transmission modes and pathways and pathogen mutation and adaptation, which can be informed by things like molecular analyses, phenotyping, and experimental studies. So next slide, please. And the next slide. So, I mean, I think we're trying to do quite a few of these things in a study we have ongoing in Cambodia, which Gavin referred to, which aims to understand how zoonotic and pandemic risk is affected by swine production systems in Cambodia and how these are configured. So we're conducting surveys across pig sector components, across smallholders, commercial farms, traders, and so on. First of all, to get an idea of how pigs move between these different types of systems, but also how the prevalence and diversity of viruses may vary between these different parts of the pig value chain. And we're also recruiting a human cohort, which will follow up over time, conducting serological and virological surveillance to get an idea of where might be hotspots for zoonotic transmission of influenza. And these data will all go into informing mathematical modeling across each of the stages that, of pandemic emergence that I referred to earlier. And next slide, please. And so I just wanted to finish by highlighting the importance of interdisciplinary research. So I focused on some of the key drivers and mechanisms by which they can impact on disease dynamics. But as highlighted in a paper by a colleague of mine, we really need to look beyond simple causal relationships between ecological drivers and transmission dynamics, to also consider the social, economic, and political context within which these things exist. So in addition to the modelers, epidemiologists, and virologists, and so on, we need social scientists to help better understand how this context is driving these profound changes, and also to evaluate the broader social and economic consequences of emerging diseases, and indeed to help identify policy options and strategies which are feasible and effective to prevent and mitigate these emerging infectious diseases. Next slide, please. So just to finally thank a few of the key collaborators and partners involved on the pig flu camp project, which I briefly mentioned earlier, which is funded by the US Defense Threat Reduction Agency. And yeah, that's it for me. So thank you very much for listening.
Thank you very much, James. We're going to have questions at the end of the session, and please feel free to post your questions via the Zoom chat box at any time, and we'll get to them at the end of the presentation. So, presentations. So please just give me a second to share my screen. Okay, so James has given us a great summary of the epidemiological and environmental factors that contribute to pandemic emergence. So I'm going to be talking about the role of evolutionary factors in pandemic emergence. And very briefly, hopefully very briefly, I'm going to talk about how viruses evolve and what viruses need or do to successfully jump to a new host, and also a little bit on how genomic data can inform risk assessment. I thought there's a lot of commentary around the COVID-19 pandemic about virus evolution, mutation, and quite frankly, a lot of it is ill-informed. So I thought that I'd spend a little bit of time just explaining how virus evolution works. So the key feature of viruses, particularly RNA viruses, is just how error-prone their replication is. If you look at a comparison with humans, there's very few mutations per replication cycle of the genome. So 3 mutations out of a genome of 3.3 billion base pairs. But for RNA viruses, we have a rate that's much higher for a much smaller genome. So you have 1 mutation per genome for each genome position for every virus that is produced during replication, and also very small genome size. So what this results in is a very high level of diversity within a single infection, and a highly diverse virus population within a single infected individual, if that is human or animal. Now, this provides a lot of variation for natural selection to work on. I just want to provide a very simple example of natural selection related to drug resistance. So we have a natural amount of variation before selection. The selective environment factor is applied, and in this case it would be antibiotics or antivirals. And then you end up with a selection producing this final population, and this phenotype is a resistance to the drug. So you have drugs applied, the susceptible pathogens are removed from the population, and you're left with this resistant population. So natural selection acts upon the virus phenotype. And then how does it work? There's people always talking about a natural selection. So this is typically what we're looking at when we're looking at virus genomes. We make an alignment, we sequence the virus, we look at the genome, and then we're looking for these differences. So here we have everything is very much the same, and then we have this one position where there's a little bit of variation. Now, this mutation or substitution can have one of 2 effects. It can produce effectively no effect in the protein, in the amino acid, where it's called a synonymous mutation. So even though there is the mutation, it doesn't change the amino acid. Or if it's in a slightly different position, it could change the amino acid, at which point it's a non-synonymous mutation. And it's the differences between the ratio of the synonymous and non-synonymous mutations that we use to calculate a selection pressure. And basically, if you have more of these mutations that are resulting in changes in amino acid compared to the mutations that don't change the protein composition, then you have a positive selection, right? So we have a neutral selection where these 2 values are equal. We have a negative selection where there's more of the mutations that result in no change. And then we have positive selection where we have more of the mutations that produce a change in the amino acid and the resulting proteins. And what are the effects of these? Well, across a genome, we don't have equal application of selection pressures. So this is a schematic of the SARS-CoV-2 genome, a very basic one. On the right-hand side, we have the spike protein. So this is the major protein that interacts with the human host. It's involved with cell attachment and also with the immune response. And it's subject to very strong positive selection because it is involved in escaping the immune response of the human host or other hosts. And it tends to be highly variable. And this is subject to a positive selection. So positive selection is also referred to as a diversifying selection because you see a lot of changes, a lot of amino acid changes being fixed into the virus population, right? And then if you look at the ORF1ab of the virus, this is a polyprotein that produces the genes involved with replication of the virus, and it's essential for the virus survival. So you tend to see less mutations there because you can't have, there's a risk for that if you have a mutation, that you'll impair the ability of the virus to replicate. And so selection favors conservation in this protein. But effectively what happens is, with the viruses, they produce a high amount of diversity when they're replicating. Not all of those viruses that will be virus particles that will be produced will actually be able to grow. But because you have such a high diversity of viruses being produced, sorry, we can ignore that one. Because you have such a high diversity of viruses being produced, it increases the likelihood that within that infected individual, for example, that you have an individual that is able to evade the immune response, or an individual virus that is able to evade a drug or vaccine-induced immunity as well. And there's also an aspect of changed environmental conditions, which is important when it comes to the emergence of pandemic viruses. So there's often a misconception that the virus population, changes in response to the a drug, right? So the virus, a single homogeneous virus population comes up against a drug, and then after coming up against the drug in the next round of replication, the virus mutates so that it can escape. It's not that. The drug acts as a barrier, and there's already some viruses that are able to escape it, and those escape through a gap in that barrier because they have pre-existing mutations that aren't affected, and then they're the ones that go on to effectively establish the infection. So how does this matter for pandemic emergence, right? How it matters is that partly addressed by James, is that these viruses that cause pandemics come from these wild animal hosts. They frequently move through intermediate hosts before they come into the human population. But a shared feature of these viruses is these multiple host jumps. And so it's this cross-species transmission introduces a lot of changes in the environment that the virus have to try and survive, because there's differences in the receptor binding of the viruses, there's differences in the intracellular conditions. So viruses have, different organisms, of course, have different temperatures, they have different receptors and so forth. So what are some examples of this? So different hosts, we have a different environment for the virus to try and replicate. So a very well-known example is the influenza PB2 gene has a mutation that is a major determinant of the adaptation of avian virus, an avian influenza virus, to humans. And how this works is that it allows the virus to replicate efficiently in the upper respiratory tract at humans. It affects the polymerase activity at different temperatures, because in the upper respiratory tract of the human is relatively, sorry, if everybody could mute, it's relatively cooler than the lower respiratory tract within the human, but it's also a different temperature to the replication site in chicken, which is often where the viruses, influenza viruses that infect humans come from chicken populations. So again, it's this high diversity of the virus population that allows the rapid adaptation to different hosts and different host environment, and the ability to overcome the challenges represented by these species jumps. Another example of this was with the 2009 swine influenza pandemic, the H1N1 virus. We saw this where in the early period of the virus, when it first emerged in 2009 and 2010, the majority of the selective pressures in the virus were driven by adaptation to the human host. So we saw mutations occurring in the polymerase genes that were directed at optimizing the efficiency of the virus replication in the human host compared to the pig. But then after a while, and in this early period, there was very little selection pressures that were immune driven. You didn't see a lot of selection occurring in the surface proteins, the hemagglutinin that is the main target of the human immune response for influenza infection. But then after a period of 12 to 18 months, you start to see a change in where selective pressures are occurring in the influenza genome. And in that period, you stop seeing selection pressures in the polymerase genes, and you started seeing directed at the surface proteins, and in particular at known immune epitopes within the hemagglutinin protein. So it's this, just to reiterate, it's this high diversity and the rapid generation of this diversity that allows these viruses to be very effective when it comes to jumping between hosts, but also in adapting to the new host so that they're able to establish themselves. So how does this help us? Well, currently, the genomic information, particularly with something like influenza, much less when it comes to other pathogens because they haven't been studied as extensively. But a lot of the mutations and genomic data is used as a shorthand for a virus phenotype. So there's been some experiments done looking at how to make viruses, avian viruses such as H5N1 influenza, transmissible in mammals. And so these experiments were effectively a biological risk assessment where you grow the virus in different hosts to look at what changes occur. And then when you identify these changes, you're able to manipulate the virus to see which of the mutations that are observed experimentally, what effect they're having on the virus biology. And this led to a number of mutations in the genome being identified that were critical for the emergence or the potential emergence of a human adapted virus. And it was a surprisingly small number of mutations, of 5 or 6 mutations. So this information and the genomic information has been put together as part of pandemic risk assessment tools There was a number of these, but the more accepted risk assessment has been set out by the WHO, where they try to look at the different types of factors that that if possessed by a virus could be or indicate that there's a pandemic risk. There's a lot of influenza viruses, there's a lot of exposure, but there's very few that are successfully emerge into the human populations. So these type of risk assessments are done all the time. And just to give one example, there was recently, well, not that recently, I suppose, but in the last 4 to 5 years, there was the emergence of canine H3 influenza A viruses, first in East Asia, but then the virus spread to dogs in the United States, or it was imported with dogs to the United States, and then started to spread there. Now, when we have a new virus that's coming up in a new host, it causes concern about the potential for a pandemic emergence, particularly with something that's in companion animals. So there was recently, during this pandemic, some evidence that cats were susceptible to the COVID-19 virus, and that's a cause for concern because humans live in very close association with companion animals, and there's a high risk of spillover. So a group that I was involved with conducted a series of experiments to try and inform the different categories that are included in the influenza pandemic risk assessment, to see whether or not these viruses, or how much of a concern these viruses would be. And the type of things that they look at are the receptor binding property. So at a very simple level, are these viruses able to attach themselves to cells in the human respiratory tract? We also look at whether or not there's transmission in different animal models, so primarily in mammalian animal models. And part of that is to test whether or not it's direct contact or whether or not it's caused by respiratory droplet, etc. And a lot of these factors have been hotly debated when it comes to the COVID-19 pandemic, because we don't have this type of information. But for influenza viruses, because they've been identified as a risk, they, at least for some of them, these type of information is collected. Now, when it comes to the genome characteristics, we're looking for molecular signatures of human infection and disease, and an example of that is that PB2 mutation that affects the replication efficiency of the influenza virus that I mentioned. And there's a bunch of other molecular signatures that are associated with pathogenicity in mammals, as well as the ability to infect, the ability to utilize the human receptor type. And we also look at whether or not there's population immunity. So do we have cross-reactive antibodies, and also susceptibility to antiviral treatment that can also be determined, at least in part, from looking at the genomic composition of the viruses. So in closing, I'd just like to highlight that these viruses that are able to mutate very quickly, generate huge amounts of diversity in a very short period of time, are well adapted to jumping between different species, and it's this ability that is closely associated with the emergence of pandemics. there are ways that we can conduct risk assessments on the actual viruses themselves to try and identify which ones are of concern. But this can't be done in isolation. It needs to be also taken into consideration the various aspects that James was talking about, such as human exposure and the ability or the opportunities to actually jump over. And with that, I'll finish, and both James and I will take any questions. And I'll stop sharing my screen. Okay. So we have some questions, if you just give me a second to find them, that were sent earlier. The first question is for you, James. So intensive agriculture, land use change and the wildlife trade have all been linked to emerging infectious diseases. Since these issues are very difficult to address, what are the best remaining options for containing or even preventing future pandemics?
Hi, yeah, it's a really good question, and I think there are clearly no easy solutions. Although I think, you know, things like wildlife trade and livestock intensification are difficult to address. So it's not to say that there aren't things that we can do to try and, you know, increase enforcement of restrictions on things like livestock, wildlife trade, but also thinking about ways in which we might be able to reconfigure even intensive livestock systems to make them, you know, improve biosecurity and reduce the movement of animals between different systems, for example. So I would say that there's still scope to, you know, certainly to reduce risk in relation to those drivers. But I guess one of the other key things in our weaponry is trying to be able to detect these things as early as possible. I guess one thing, I think surveillance is going to be a topic of the next webinar, but one thing I didn't really talk about was the role of big data. So this is of course being used for whole genome sequencing, but also the role of big data for surveillance more generally. For example, aggregating news reports from the internet, search engine queries, social media, smartphone data, retail records from pharmacies, even satellite and remote sensing data, all potentially to improve the timeliness and geographic resolution of information and help address sort of gaps in existing surveillance programs, and also provide evidence to better inform predictive models and planning for disease management.
Okay, thanks, James. So another question that was sent in previously was, why was SARS brought under control with a small number of infections, but COVID-19 was not? I can address this. It's not exactly my area of expertise, but I think part of the reason is because with SARS-1, people got, there was very little asymptomatic transmission, and people also got very ill. So it was more effective. Quarantine was more effective because there was very little asymptomatic transmission. Whereas with this virus, which is also similar to influenza, there's a period where there's asymptomatic transmission, which means that once you've identified an infected individual, that quarantine is going to be less effective. And a related question for you, James, to the earlier question was, how might we prevent disease spillover for the sake of human health, and how can this tie into environmental and climate goals?
Yeah, again, that's a really good question, and I think there's lots of synergies between measures to reduce spillover and also to achieve climate change and environmental goals. So, I mean, this includes things like, as I was discussing, rethinking our food and livestock production systems, ways of reducing meat demand to make these systems more sustainable and environmentally friendly. So biodiversity loss and land use change can also increase risk of spillover. So measures for sort of tackling these things, and then things like deforestation can work to achieve both goals. And then, of course, thinking about the huge rise in international travel and how this increases the risk of new diseases spreading very quickly. Again, thinking about measures to reduce this. For example, I think everyone is kind of getting more experience with virtual conferences and meetings, and potentially these kind of things can also play a role in both things.
Okay, and just while we're discussing that, there was another question from Gary Ong that was any way that we can work with global or multilateral organizations to detect or perform surveillance in high-risk regions or areas. And the comment from Gary that this is intensive, but compared to the economic and health impacts, perhaps there's a good trade-off. So James, perhaps you could talk about the sort of partly the difficulties in setting up these type of surveillance that you talked about with the swine project in particular.
Yeah, I mean, I think my experience isn't so much, I guess, with kind of multilateral surveillance systems across countries, but at least even just within countries and particularly developing countries, combining, the interface between human and animal health sectors and their surveillance systems is quite challenging. So these kind of sectors previously operated in isolation, I think. So that's certainly a challenge for surveillance, and also for One Health focused research projects. But I think at least the increased kind of appreciation of the importance of One Health approaches is gradually improving this.
But it also hasn't been a very active area for securing research funding. I mean, it can be quite tough.
Yeah, definitely. I think funders have also they're increasingly supporting cross-sectoral and interdisciplinary projects, but again that can lead to problems in the review process in terms of which experts review proposals and that kind of thing. Yeah.
That I should also mention that the third webinar in this series will also deal with global and multilateral organizations and what role that they can have or have had as well in pandemic and pandemic prevention. So just moving on, so there was a question from Erica about detection of positive selection with COVID-19 so far, because it's quite early on, and also whether or not, because the virus is widespread, if we would see the emergence of different strains that could cause a problem when it comes to vaccine, because the hope is to have a vaccine that works universally, if you like. And there was also a related question from Ning Mao, where I had mentioned that the spike protein was variable, and she was just mentioning that she'd seen news saying that it was relatively conserved, and the effect that could have on vaccine as well. So I think, we're still only 6 months in. When a new virus emerges into a human population, there's not very much pre-existing immunity for a pandemic. If there was a lot of pre-existing immunity, we would have been exposed to something previously. So what that allows is for a lot of an increase in diversity of the virus, because it's spreading through susceptible hosts, right? And there's no antibodies that from previous infection. But a lot of the mutations aren't very informative, and it takes sort of about 6 to 12 months of having the virus circulate widely before you would start to see true sort of diversification and selective pressures, at least going on influenza. This virus mutates a little bit more slowly, so the time frame could change. When it comes to the spike protein, the spike protein in this particular virus is very conserved, but again, we're only 6 months in Oh, sorry, I have to check myself when I say only 6 months in, because it feels like it's been going on longer. So the spike protein, yes, it is very conserved. At the moment, when the 2009 H1 influenza emerged, the vaccine component for that virus was not updated for, I think, about 3 or 4 years. And that was largely because in the first 18 months to 2 years, there was very little evolution of the virus going on, because there was very little, it takes time for infection to spread in the community. So relatively, the spike protein can be subject to more change than the other proteins, but at the moment it's not. I hope I've answered those two sufficiently. And there was another question also from Erica about, we know how these pandemics start in general and the importance of the ecological aspects. Why is there little focus on avoiding a spillover at the first place and more on the vaccines and medicine development? I can start with that, and then James, you could chip in. So effectively, our surveillance and warning system at the moment is using humans as sentinels. So in farms where they have vaccine preventable diseases, they'll often have what they call a sentinel animal. So a sentinel chicken, or chickens that you don't vaccinate and the rest of the flocks are vaccinated. And then, so if those animals get sick, then you know that that there's something going wrong. And that's currently, as far as I can tell, is how pandemic emergence in humans works at the moment. We have surveillance for unusual diseases emerging in humans. I honestly don't know if we could do better than that. James, do you have anything to add?
No, I don't have that much to add. I mean, I think, yeah, I guess it's also a question of scale. So it's very difficult to implement large scale surveillance across, all kind of livestock or potential reservoir animal hosts. So although I think we can do more to understand these dynamics to inform kind of risk-based surveillance approaches, it's just really difficult to predict, of course, when or where a disease spillover event might then go on to cause a pandemic. So as you say, using kind of humans as sentinel hosts is kind of something that we have to rely on to some extent. Yeah. Also, I think, there's a lot of these probably risky settings and practices associated with spillover. They're also kind of deeply ingrained within culture and socioeconomic factors of certain settings. So those things can also be very difficult to tackle as well.
Yeah, so I think, one way potentially of improving our chance of picking something up is, strengthening healthcare systems in areas where you do have risk of emergence, so that there can be earlier detection and things like that. So some more sort of vaccine related question. There was one from JJ Go about whether or not there's a possibility of a universal vaccine anytime soon. And then another one from Shi Tio asking about, because the virus is able to mutate, then a vaccine would create a massive selective pressure for mutations, so that you would lead to some sort of vaccine escape. And then if that means that the vaccine is unlikely to be effective. So honestly, so there's going to be a number of different targets, I suppose, for the vaccine. There's always the possibility of vaccine escape due to mutation. But even with influenza viruses, the vaccine composition is updated every 6 months, but there's 4 different components in that vaccine. There's 2 influenza B viruses and H1 and H3 virus. Each of those components is updated usually every 2 to 4 years, depending on which component. So this virus, the SARS-CoV-2, the mutation rate is lesser than we have for influenza. So even with the mutation and the selective pressure that a widespread vaccine campaign could induce, I don't think that you would necessarily see a complete failure of the vaccine. And it's possible, if that did happen, if there was escape, that it would just be a matter of updating for the new virus, if you like, or the novel strain that had emerged, similar to what we do for influenza vaccine. And just as my personal opinion, unless we have a vaccine that is able to completely prevent infection, and there's some sort of global campaign, that could be the situation that we end up in, where we have a vaccine that needs to be updated on some sort of regular basis. But that's entirely speculation and personal opinion, just let me state. It seems, I think, that we're out of questions, and we're also out of time. I would like to thank everybody for attending. I hope it was informative. We have the next talk, maybe Amina or somebody else from Global Health Institute could briefly provide some information on the next talk. But
sure. Should I jump in here?
Yes, yes, please.
First of all, I just want to say to James and Gavin, thank you for really excellent, informative presentations. This was exactly the idea, and maybe someone had mentioned it, from moving the focus away from just how are we managing the current situation to really gaining understanding of why these emerge. And as the series we look at a few more questions around that. So the next one is a week later. Same time, same place. And we sent out the registration email. You do have to register for it separately. So you do need to go into that email and find the registration link and register for that. And the third one, similarly, we'll send it out a week later and you will register for that separately. And some of the questions, especially the kind of large questions of, the second one will be looking more at surveillance and looking at how really these things are managed. And then the third one will be really looking at what maybe we can, using this information, what we can do to prevent it, and how can we bring all our many global partners and international institutions and the multidisciplinary disciplines together to do something about it. At least that's the hope. Thank you again, Gavin and James.
Yeah, so I'd also like to express my thanks to James for joining and for providing such an informative talk. Thank you very much.
No, thank you very much for inviting me. Yeah, it was a real pleasure, and I'm just glad I was able to connect in the end.
Okay, so I think we can end the meeting there. Thank you all, and hopefully we'll see some of you next week.